guys, welcome back to part 2 of our Kcse Biology part 2 2021, and we start with the question 6. Previously we had looked at question 1 up to 5, which entails 40 marks each question, 8 marks. Let's look at section B, which contains the graph and the essay questions. Answer question 6 and either question 7 or 8 in the spaces provided after question 8. Question 6, the data below shows the average number of ticks per animal in a certain farm before and after spraying the animals with a certain chemical. The spraying was done once every month. The data was tabulated as shown below. Time in month, 0 month, 2, 4, 6, 8, 10, 12, 14. Average number of ticks, you can see 200, 90, 40, 20, 16, 25, 40, 45, sorry, 90. Before we look at the graph, draw the graph or explain the graph, as you can see, when the time in month is increasing, the average number of ticks are reducing. You can see 200, 90, 40, 20, 16. Then at the 10th month, it starts to increase. 12th month also increases, then 14th month increases further. Plot a graph of number of ticks against time, 6 marks. And in drawing the graph, you must label the axis, and we have our y-axis where we have average number of ticks, must label it. Then at the x-axis we have time in month. And another key point or important point when drawing a graph, it must have an origin. The y origin, which is here, 0, it means starting from 0. Then the x origin, which is starting from 0. Then the scale. The scale should be workable, not just any scale. And when you look at our scale, 2, or you can say in other terms 2 months, it is represented by how many boxes? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It means 10 boxes represent 2 months. And it is easier to work with that. It means 5 boxes represent 1 month. Then on the y-axis, you can see we have a scale which shows 10 boxes representing 40 ticks. It means 5 boxes will represent 20 ticks. Then when drawing the graph, you should use a freehand, don't use a ruler. You plot those small x's which will guide you in plotting a smooth graph in order for you to get the 6 marks given here. Let's look at the questions. Part B. Account for the shape of the graph between 0 and 8 months. You can see the 0 and 8 month. You can see the average number of ticks is decreasing as you move towards the 8th month. Generally, we can say we have a rapid decrease in number of ticks as you move towards the 8th month. And in accounting, you say the observation and then we explain why. So you have seen there is a decrease in the number of ticks. The chemical was poisonous, killing the ticks. The ticks had not also adapted to the chemical or developed resistance. Then Roman 2, 10 to 14th month. You can look at the 10th to 14th month. You can see the average number of ticks is increasing. You can see the number of ticks per animal increased because the ticks had adapted to the chemical or had developed resistance. And the resistant ticks produce enzymes that made the chemical harmless to them. See from the graph, determine the average number of ticks after spraying the animals for 5 months. You need to look at it here. The 5th month is here. Then you can approximate here. This is where we'll have to look for the average number of ticks, and at this point 1, 2, that is the second box after the 20 number of ticks here, and that is around 28 ticks. Can I give a benefit of doubt plus or -2? Then if the animals were allowed to graze in an open field, construct a food chain with 5 organisms in which ticks are secondary consumers. The organisms you have: grass, animal, tick, bird, vulture. And the correct or specificness of organisms in the food chain should be identified. Producer, we give a mark. Then the energy flow from the producer, which is grass, to the consumers. And the organisms present should be 5. That is 1, 2, 3, 4, 5, in order for you to get the 4 marks. Then part E states 3 methods by which average number of ticks per animal could have been estimated. And these are estimated by mapping based on the various parts of the animal body, or a physical counting or total count of census, or sampling of animals. Let's look at question 7, which is an easy question. Part A, explain the role of liver in blood sugar regulation. Under the role of liver in blood sugar regulation, we talk about when the blood sugar level are above the normal or below the normal, what happens. When blood sugar level is high, above the normal, insulin hormone is produced by the pancreas, and it stimulates the liver cells to convert excess glucose to glycogen. Remember, you must say excess because the glucose levels are above the normal, or some excess glucose is converted to fats. Then when blood sugar level is below the normal, glucagon hormone is secreted by the pancreas, stimulating liver cells to convert the glycogen stored or fat stored back to glucose. Then part B, describe how human blood is adapted to its function. And the human blood, remember, it contains the liquid part of blood, or the liquid part, which is blood plasma, and plus the cells, which are the blood cells, red blood cells, white blood cells, and the platelets. Plasma, that is the first component, saying how they are adapted. And adaptation involves the structure, modification, and function. And we have the plasma, fluid part of blood consisting of dissolved and undissolved substances. And the plasma acts as a medium in which various substances are transported in the body. It also acts as a medium in which various metabolic reactions occur and plays a role in thermoregulation by distributing heat around the body. Next, we have platelets. They contain proteins that help in blood clotting, thus preventing excessive loss of blood. Excess must be there. Then also prevent entry of pathogens. And the protein here we're talking about involved in blood clotting have a thrombokinase or the thromboplastin. Then the white blood cells are irregular or amoebic in shape, and they protect the body against attack by pathogens by engulfing the pathogens and also releasing antibodies against the pathogens. They are also numerous to enhance the body defense mechanism. Then in case there's a named white blood cells, the monocyte, lymphocyte, or granulocytes should be accepted, or the lymphocytes are granulocytes producing antibodies, and the granulocytes, phagocytes, neutrocytes by engulfing be given a mark. Then red blood cells are biconcave in shape to increase surface area for diffusion of gases or squeezing through the blood capillaries. They lack nucleus to allow for packing of more, who must be there, more hemoglobin. Then they have hemoglobin that has high affinity of oxygen. They are also numerous to increase surface area for transport of more oxygen, more must be there. Then it has carbonic anhydrase enzyme for faster loading and offloading of carbon dioxide. And the last question, which is Kcse question 8, part A, explain how presence of chloroplasts in gut cells affect the opening of stomata. Let's look at it. Chloroplasts are site for photosynthesis. During the day, photosynthesis takes place. Glucose is manufactured. Glucose being osmotically active increases internal concentration of the gut cells. Then water is drawn into the gut cells. Gut cells become turgid, bulging outward. Stroke an equal expansion of the gut cell results in the opening of the stomata. Then you can also use the starch interconversion theory. Chloroplasts are sites for photosynthesis. During the day, photosynthesis takes place using carbon dioxide, making the pH rise in the gut cell, favoring the conversion of starch into glucose. Then this will affect the concentration, the gut cells, thus leading to the opening of the stomata. Potassium theory can also be used, and this talks about the ATP accumulating in the gut cells, making the gut cells to draw potassium ions. Then probably end up causing higher concentration in the gut cells, and this causes bulging or unequal curvature of the gut cells, resulting into opening of stomata. Then the last bit, that is describe how various environmental factors affect the rate of photosynthesis, and they are specific to environmental factors. Number one, you have carbon dioxide concentration. Concentration should be there, and remember, carbon dioxide is a raw material for photosynthesis, and an increase in the carbon dioxide leads to an increase in the rate of photosynthesis, and that is up to a given optimum. Beyond optimum, the rate of photosynthesis remains constant due to other limiting factors. For example, light intensity, it can be water, it can be temperature. The next factor, we have light intensity or quality of light. And light provides the energy required for photosynthesis or photolysis of the light stage, and the rate of photosynthesis increases as light intensity increases up to an optimum level, and beyond the optimum level, the rate of photosynthesis remains constant, and that is due to other limiting factors, and the rate of photosynthesis drops. The next we have temperature, and remember, photosynthesis is affected by enzymes, or the enzymes are involved in photosynthesis, and temperature always affects the enzymes. In this case, low temperatures inactivate enzymes, thus reducing the rate of photosynthesis. And as temperature increases, the rate of photosynthesis increases up to an optimum point, and high temperatures above the optimum denature enzymes, reducing the rate of photosynthesis. Next we have water. Water is a raw material of photosynthesis. Remember, it is broken down to produce hydrogen ions and oxygen. It influences the opening and closure of stomata, which in turn affects the diffusion of carbon dioxide into the leaf. And that is the end of our revision of paper 2, Kcse Biology 2021. Please like, comment, share, subscribe, and ask any question, and stick to the channel for more revision series and other Kcse questions in preparation for your Kcse exams. Thank you.